Assalamu alaikum. My name is Muhammad Afaq Qureshi, EP1770096, and my topic is Morris Water Maze Experiment. And the portion that I have been assigned is discussion. Okay. Uh, this experiment was developed by Morris, and it can be altered in numerous ways to investigate different types of memory. So basically, this experiment deals with types of memory. For example, working memory, reference memory, task strategy. Task strategy is basically not a type of memory, but the strategy that is developed to complete any task is based on the memory or the learned things that you have in your mind. The procedure contains two critical variables which represent a deviation from the other versions. And these are pre-training and testing on a single day. Pre-training, as it is, you can assume by the name pre-training, is that pre-training the hippocampus is involved in spatial memory, uh, spatial relational memory. Okay. The water maze basically test spatial memory. The Morris water maze test. However, there are numerous other components of the task that do not involve spatial memory. For example, the stress involved with the task, understanding of the rules, learned helplessness, and these are there are also other factors. The three pre-training trials teach the animal about these properties of the task, which are that they should have to be on the platform to be rescued. And if they are not able to find the platform, then they will have to wait till someone picks them up and uh, places them on the platform. And that there is no escape from the maze, and the only escape is that they have to find the platform. Okay. So for this, the rodents are pre-trained uh, in mode of trials, and then the experiment is performed to find out their behavior. Okay, now testing on a single day is basically developed for female rodents, humans, primates, because of their cyclic changes in steroid hormone levels, and these steroid hormone levels are progesterone and estradiol. Okay, these two hormones affect the result of our experiment by altering the hippocampal anatomy and hippocampal cell function. Okay, uh, so that is why to eliminate these changes in the result, we have to perform the whole experiment or the total all of the trials in a single day to exclude or to eliminate the changes that will occur by these hormonal changes. Okay, my research topic is quantification of swim patterns in the Morris water maze. Okay, by Sergio Glutendorst, Ron, and Melly. Okay, this article is basically telling us that there are not only two types of swim patterns, but three type of patterns. Okay, using the traditional parameters, we have only two swim patterns. One is the spatial strategy, which is uh, from spatial memory, and the other is random strategy, random swim pattern, or random strategy that occurs without the setting of any goal. Okay, so now let's continue with the article. Spatial learning and memory in rodents is most often assessed in the Morris water maze. Neurobiologists have to distinguish behavioral patterns to unravel underlying neuronal systems. Okay, uh, behavioral biologists are confronted with the problem of how learning and memory can reliably quantified using behavioral measures. So, Morris water maze is an experiment which is widely used for the assessment of spatial memory. And in this experiment, or in this article, we are basically uh, working on the different types or different swim patterns to know that there are not only two parameters or not only two types of behaviors. That is one a special strategy and the other is random, we are going to uh, study deeply the spatial strategy and find out if there is anything different from spatial memory or spatial strategy or not. Okay, now, for example, animals that direct their search predominantly to the area of the maze where the platform was located during training can have same latency. Okay, now there are some factors on which I'm not going to explain the procedure of the Morris water maze. Okay, so the thing is that we are going to videotape the behavior of the mice, rodents, and study their swim patterns on the basis of some parameters which are velocity, latency to platform, total distance swum, time spent in quadrants, crossing of platform locations. These are, and the last one is cumulative distance to zone. We are going to integrate this factor into our research data and find out if there is anything new or not. Okay, the aim of this study, the aim of the present study was to identify and distinguish different search strategies of animals using cumulative distance of two platform parameter. Okay, in this article we are going to identify and distinguish between different search strategies of animals by using cumulative distance to platform parameter. Okay, conventional parameters were calculated as well. Furthermore, we were interested to see whether explorative behavior. Okay, in our sample of swim patterns, we have characterized three different types: spatial, concentric, and random swim. We have three different types of swim patterns, and these are spatial, concentric, and random. Okay, now let's move on to method. Method is also method or procedure that I'm not going to discuss it here. It is about the length of the diameter and depth of the maze and how much you should fill water and the ratio of the platform and how we are going to take different trials. For example, here, table number one, path length in meters and velocity during two free swim trials. Okay, these are the search patterns and path length in meters. And path length is the distance that is swum by the mice. Okay, FS1 is free swimming trial 1 and FS2 is free swimming trial 2. And this is velocity in centimeters per second, free swimming trial 1 and free swimming trial 2. As you can see that the velocity in free swimming trial 2 is increased with respect to free swimming trials 1. Okay, every trial is performed with a 5-minute gap, and the time interval between two different set of trials is 60 minutes. Okay, swim patterns of these two free swim trials were subjected to further analysis. FS1 yielded information on the behavioral response involved in situation. FS2 yielded information on learned behavior, that is the strategy used. Okay, for data analysis, we are going to videotape all of the trials and then we are going to put this data in this system, which is analysis system EthoVision 1.7. And then we are going to analyze the data, and also we are going to integrate cumulative distance to platform, which is a parameter which is not used traditionally, but we are going to use it to find out if there is any other type of behavior from which we have studied in traditional parameters. To allow comparison between experiments with different sampling steps or measures, we presented cumulative distance to platform as a percentage. This was done easily on the basis of the following calculations. Okay, now we have performed our experiments and then we have our data, and we are on the basis of this data, we have drawn some histograms, bar charts, and graphs, and now we are going to study those. Okay, this is the video taped path length or the of the mice, as you can see, there are three different types of patterns, as you can see, in spatial, non-spatial, concentric, and non-spatial random. Okay, in spatial, as you can see that the whole trajectory or whole distance or of the mice is in position platform and in platform position or platform quadrant. And in non-spatial concentric, you can see that the mice is moving around the wall of the maze. And in random, you do not have any predictive or any pattern that you can understand. Okay, now in a spatial, the mice is goal directed, as the mice knows that there is a platform in the maze, and the mice is trying to find out that pattern, and on the basis of his spatial memory, he is directing his search towards the platform quadrant, because somehow the mice knows that the platform quadrant is somewhere in this quadrant, and it is goal directed. Okay, reference to platform location, latency is less than 10 seconds, high crossing number, distance to platform is less than 20, and time spent is less than 40 seconds. In concentric, it is semi-goal directed. Uh, in this type of trials, the mice is directed to some extent towards the platform, and he knows that the platform is somewhere located towards the wall of the maze. Okay, semi-goal directed, reference to platform location, latency is uh, less than 10 seconds. Okay, high crossing number, and distance to platform is between 20 to 23. And random, it is not goal directed. The In the random trials, the mice does not know that there is any type of platform, and he does not know that the only escape route is finding out the platform. Okay, so latency is obviously greater than 10 seconds, low crossing number, and distance to platform is less than 23. Okay, reference to platform quadrant, time spent, and the time spent here is greater than 30 seconds. Okay, now distance to train platform location and time spent in platform quadrant. As you can see here in the persistent strategy, the time spent in the platform quadrant is higher, and it is decreasing in a linear fashion, as the mice are now incorporating their memory, which is gained from the trials, and now they are spending less and lesser time in the platform quadrant, because now they are finding the platform fastly. And in concentric, as you can see, the again the time is decreasing, and in random the time is also decreasing linearly. But the distance to trained platform location is higher in random, because the mouse is now traveling a large distance and is moving towards other quadrants, and is um, in a random fashion to find out the platform. In concentric, it is moving in circles. Okay, now data preparation. To circumvent possible interference with general activity, we chose swim patterns of 24 mice with similar path lengths during pre-swimming trials one. And now we have incorporated this data into analysis method ANOVA, which is analysis of variance, and we have some bar charts that we are going to discuss now. Okay, now we have A, which is seconds. Okay, latency to A, and latency in seconds, as you can see, persistent, concentric, random, and free swimming trial one was performed before the training of the rodents, and free swimming trial two is performed after the training trials. So you can see that there is a major difference between FS1 and FS2. So this black area is the platform quadrant. Okay. And in persistent, you can see that in a free swimming trial too, that the time spent in the platform coordinate is lesser because the mice knows that there is a platform and he is trying to search or locate that platform. And in concentric, as you can see, it is greater than persistent but lesser than random, because in random it is not goal directed, and the mice does not know that there is an any platform here. Okay, crossings of the crossing here is crossing of platform quadrant. So you can see here that the crossing here are very less, because the mice does not know if there is any platform or not. But as in pre-swimming trials 2, you can see that the crossing of platform quadrant is huge and a major difference from free swimming trials 1 and free swimming trial 2, and the same fashion is exhibited in free swimming trials 2. Okay, now second, a percentage to cumulative distance, and the cumulative distance in persistent, concentric and random here is this, before training and after training is this. Okay, and now time, percentage of time spent in quadrants of animal with persistent, concentric and random swim patterns during the free swim trials before FS1 and after FS2 training. Okay, the circular insert represents the division of the pool to four. The black area is the platform location, and the black bars denote the platform quadrant, and the other patterns refer to the location adjacent or opposite to the platform. So as you can see here, that the time spent in the platform quadrant is lesser in persistent, and in free swimming trials too, the major time spent is in the platform quadrant. But in random, no such fashion was observed. Okay. Now, results using the traditional parameters, we classified two distinctly different patterns, a persistent strategy or a spatial strategy and a random strategy. But with the incorporation of cumulative distance to zone, revealed a third pattern, which is concentric pattern. In traditional parameters, the concentric strategy was hidden under the spatial strategy, but with the incorporation of integration of cumulative distance to zone, a new strategy comes up, which is concentric strategy. Okay, now the concentric pattern is different from the one seen in animals with hippocampal lesion. So the concentric strategy or concentric pattern is different in those animals which have hippocampal lesions, because hippocampal is the area of the brain which is responsible for the spatial memory, and if anything happens to the hippocampus, then the spatial memory of that mice is altered or changed or somehow affected. So those mice will not be able to perform or will not be able to do spatial learning. So this was the difference between spatial and concentric strategies. Okay. In traditional parameters, the concentric patterns or the concentric strategy was in spatial strategy. So it will give a false result to some extent, that the mice that cannot perform or are not able to do spatial learning, they would show concentric strategy. But due to traditional parameters, they were coming under the spatial strategy. Okay, now and represent spatial persistent mice. A significant negative correlation was found between cumulative distance to platform and time spent in the platform quadrant. For concentric group, no significant correlations were found, and random patterns revealed significant correlations between cumulative distance to platform and the actual length of the swim path in the platform quadrant. So this is a major difference between these three different strategies that were uh, performed by the mice during the experiment. In addition to the correlation between cumulative distance to platform and time spent in the platform quadrant, now table 2, latency to find the platform during training trials with platform for animals with spatial and concentric and random swim patterns. Okay, this experiment was performed on 3 consecutive days, and on the first day, a set of 3 trials was performed with a gap between these 3 trials was 5 minutes, and then on second day, 4 set of trials were performed, and then there was a gap of 60 minutes, which is inter-trial interval, and then another block of 4 trials was performed, and on the third day, uh, again 4 trials were performed, and these were the trials that were performed before free swimming trials 1 and free swimming trials 2. So in this trials you can see that the time spent is in the platform quadrant is decreasing, and the crossing of the platform quadrant is increasing. So now we will all animals learn something. Table 1 for latency during yes, it is evident that all animals learn something because they knew that somehow there is a platform. Even in random strategy, the mice shows biasness to platform quadrant because they knew that there is a platform hidden somewhere in the water, and they have to find that platform in order to be rescued. Okay, discussion. Yeah. The present study revealed that the rarely used parameter, which is cumulative distance to platform in the analysis water maze behavior, allows a more refined distinction between swim patterns than traditional approaches. In a group of mice pre-selected on the basis of swim path length before training, 3 different patterns were detected: spatial, persistent, concentric, and random. Criteria for differentiation were given on the basis of cumulative distance to platform and time in platform quadrant. Swim patterns indicate the strategy that animal use to solve the task. Different patterns reflect different ways of information processing and thus underlying neuronal substrate. Therefore, a fine-tuned method of classifying behavior is required. When an animal moves towards a goal, such as the submerged platform in the water maze, it may do so using only distal cues or by performing a previously learned motor routine like swimming concentric circles some distance from the side wall, or by combining these 2 methods. These methods can be efficient in a number of ways, but do not require special information processing. Okay, so this is the major difference between concentric and in spatial memory, because in spatial memory the animals learned the location of the platform. Okay, again I'm repeating this. In spatial strategy, the animals learned the location of the platform. They knew that which one is the platform quadrant. But in concentric strategy, the animals did not knew that there is which one is the platform quadrant. So they started to move along the wall of the maze in order to find out the platform, because they didn't have any biasness towards the platform quadrant. They only knew that there is a platform which is near to the wall of the maze. So in order to find that out, they have to move towards the wall, and they have to swim in a circular manner in order to find out that platform. These methods can be efficient in a number of ways. Okay, the spatial strategy requires the processing of complex stimuli configuration. Animals focus their search to the trained platform location. The concentric strategy, which was found to be hidden in the spatial, persistent, a group in the present study could be detected only by the use of cumulative distance to platform parameter. Otherwise, these animals would have falsely classified as the capable of spatial learning. And this was the topic, or I would say this was the summary of my research article. And cumulative distance to platform can be calculated only by image analysis system, and we should use the advantages of automated computerized techniques of quantity to quantify behavior. Of course, we are confronted with a large amount of data, but the flexibility of the system makes it possible to extract the essentials in an efficient way. Originally, cumulative distance was performed as a measure to quantify and compare search patterns of old and young rats. Our study shows that combining cumulative distance to platform with the time spent in platform coordinates measure further improved analysis of swim patterns and thus allowed identification of more subtle differences between swim strategies. Okay, that's all from my research article. Thank you so much. Khuda Hafiz.